Hello guys, thanks for watching. Today I'm going to take you through a physiology topic. It's called the cell junctions. A cell junction is the connection that exists between neighboring cells or contact between cell and extracellular matrix. We have three major types where all the cell junctions fall, namely the occluding junctions, the communicating junctions, and the anchoring junctions. I'm going to take you through the three major types and the cell junctions that fall under each. First, we start with the occluding junctions. These are junctions between cells that prevent intercellular exchange of substances. They prevent movement of ions and molecules from one cell to another. The only example under this category is the tight junction, and we shall go into it. The tight junction, or the zonular occludens. The tight junction is an intercellular occluding junction that prevents the passage of large molecules. It forms a region where the cell membranes of the adjacent cells fuse together firmly. It is present in apical margins of epithelial and endothelial cells in the intestinal mucosa, the wall of the renal tubules, capillary wall, and the choroid plexus. Looking at the structure of the tight junction, the junction is made up of a ridge with two halves from either cell. Both halves fuse with each other very tightly and occupy the space between the two cells. The halves consist of tight junction strands. There are also proteins involved in the formation of the tight junctions, falling under two types. We have the integral membranes protein and then the scaffold proteins. These proteins are anchored in the strands of the ridge. The functions of the tight junctions. We have 1. They hold the neighboring cells of the tissues firmly and thus provide strength and stability to the cells. It also forms a selective barrier for small molecules and a total barrier for large molecules, hence the selective permeability. The tight junctions also prevent lateral movement of proteins and lipids in the cell membrane, thus acting as a fence. This maintains the different composition of proteins and lipids between the apical and the basolateral cell membrane domains. This is the fencing option, the fencing function. The fourth, the fencing function of the tight junction also maintains the cell polarity by keeping the protein in the apical regions of the cell membranes. This maintains the cell polarity. The tight junctions also in the brain capillaries form the blood-brain barrier, preventing the entrance of many substances from blood into brain tissues. The next category is the cell junction is the communicating junctions. These permit the intercellular exchange of substances, that is to say, movement of ions and molecules from one cell to another. Examples of communicating junctions include gap junctions and the chemical synapses. First, we shall talk about the gap junctions, or the nexus, in other words. The gap junction is an intracellular junction that allows passage of ions and smaller molecules between the cells. It is present in the heart and in the basal part of the epithelial cells of the intestinal mucosa. Taking to its structure of the gap junction, the channel is made from the cell membranes of both cells, connecting the cytoplasm of the two cells. So, the molecules move from one cell to another directly through the channels without having any contact with the extracellular fluid. The channel also consists of two halves, each half belonging to one of the adjacent cells. Each half is surrounded by 6 subunits of proteins which are called connexins. The diameter of each channel can be regulated by intracellular calcium ions. When the concentration of the calcium ion increases, the protein subunits of the connexin surrounding the channel come closer to each other by sliding, thus decreasing the diameter. The functions of the gap junction include permitting the passage of glucose, amino acids, ions, and other substances whose molecular weight is less than 1000. It helps in exchange of chemical messengers between the cells. It also helps in the rapid propagation of action potential from one cell to another. Another kind of communicating junction is the chemical synapse. These are junctions between nerve fibers and the muscle fibers, or they can be between two nerve fibers. And through these channels, signals are transmitted by release of chemical transmitters. As we have seen all the communicating junctions, let's go to the anchoring junction, which is the last category. The anchoring junction provide strength to cells by acting like mechanical attachments either between two cells or between a cell and then extracellular matrix. These are present in tissues like heart muscle and the epidermis of skin, which are subjected to severe mechanical stress. These junctions are classified according to the types of filaments they use. 1. We have the actin filament attachment junctions, where we have two examples: adherence junctions, which are cell-to-cell junctions, and also the focal adhesion, which are cell-to-matrix junctions. The second group is the intermediate filament attachment junctions, where we have the desmosome, which is a cell-to-cell junction, and also the hemidesmosome, which is a cell-to-matrix junction. Going to the first, which is the adherence junction. This is a cell-to-cell junction which uses actin filaments. In this junction, the membrane of the adjacent cells are held together by some transmembrane proteins called the caderins. Adherence junctions provide strong mechanical attachment of adjacent cells. They are located in the intercalated discs of the cardiac muscle and the epidermis of the skin. The second is the focal adhesion. This is the cell-to-matrix junction, which uses also the actin filaments. It is found in epithelia of various organs connecting the cells with their basal lamina. It has transmembrane proteins called the integrins. The third is the desmosome. This is a cell-to-cell junction which uses the intermediate filaments. Here, the membranes of the adjacent cells are thickened and become spot-like patches. Then the intermediate filaments are attached with the thickened patches, and the transmembrane proteins involved here are the catechols. The desmosomes are found in the epithelial linings of. The fourth is the hemidesmosome. This is a cell-to-matrix junction that uses also the intermediate filaments. This junction is like half a desmosome, and the thickening of only one cell occurs. It mostly connects cells to their basal lamina. The proteins involved in this junction are the integrins. We come to the end of this educative video. Well, I hope you liked it. Support us by sharing this video, subscribe to the channel, and like the video. Be blessed. We'll see you in another video.